Hey folks, and welcome to another edition of Zog Science. Today we're going to be talking about genomics, and we're going to talk about a lot about the Human Genome Project, kind of how that was accomplished, and then what were kind of some of the results of the project. So what is genomics? Well, genomics is the study of whole sets of genes and their interactions, and it is only possible because an entire genomes or various genomes for different organisms have been sequenced in their entirety. This is going to include the field of bioinformatics, which is the use of computers to help process and analyze different biological data, which is mostly just going to be genetic information. What is the sequence of the DNA? What proteins are produced from that DNA? What's the structure of the proteins when they fold? And then all that information gets kind of processed and analyzed and used in a wide variety of different ways. We're going to talk about some of those practical applications as well. So let's talk about the Human Genome Project and genome sequencing. The first bacterial genome was sequenced in the mid-1990s, although virus genomes had been sequenced before that. Virus genomes pretty small compared to bacteria and eukaryotes. The Human Genome Project was started in 1990 and it was a intergovernmental, which means we've got governments from across the world as well as kind of an academic consortium, and they were the ones who first kind of sunk a whole bunch of money into sequencing. There was also a private group that was competing along the way. They started in 1992, and the two competing organizations, they kind of worked together in the end to release their results. The two of them kind of both came up with good techniques and good procedures, and actually used slightly different techniques, but overall they kind of ended up at the same place. So the governmental approach to sequencing, what they would do is they would identify genetic markers and build a linkage map. And we've kind of talked about linkage maps before. And so what they did was that they were able to kind of find these genetic markers along the way. So that way they kind of had an idea of where they were on a particular chromosome. Then what they did was that they created a physical map by breaking a chromosome into fragments using restriction enzymes with overlaps. And so they would say, okay, we've got these fragments, we sequence like a small part of it and figure out, okay, where's our overlap? Okay, these ones overlap, therefore this is our physical layout of where things need to go, right? And we can use our genetic markers to help us lay that out. And then we will do DNA sequencing of those individual fragments. Once we know the sequence of the individual fragments, because we know that they go next to these two other fragments, we just kind of slot it into the middle right there. So this was a very methodical approach that yielded a little bit more, I would say, accuracy, right? A little more just accurate, right? It's going to be more. Higher fidelity. That's what I want to say. Higher fidelity. It's got a higher fidelity, more representative of the true sequence of the chromosomes of the genome. So I've got a little animation of kind of how that works.
As represented by this huge stack of paper, the human genome contains more than 3 billion nucleotides, or DNA letters. The first stage of the public Human Genome Project focused on identifying marker sequences, or unique tags, shown here in yellow, at regular intervals throughout this Book of Life. Once enough sequences were tagged, various blocks of the genome were allocated to different academic centers for sequencing. To begin the sequencing process, several copies of a section of DNA, represented here as a page of text, are cleaved to produce smaller fragments. Although it looks fairly orderly, this step is small-scale shotgun, which creates numerous random fragments. Each fragment is sequenced. Then computer programs align the overlap between fragments to build up an entire page. Marker sequences, shown in yellow, help establish the order of pages in the Book of Life. This methodical process produced huge amounts of data that have been used to virtually reassemble our genome. However, there are gaps. Repeat sequences are common in the human genome, so repeats from entirely different chromosome regions may be erroneously joined together. It will take many years to detect mismatches caused by the repeat sequences. Some regions, especially near the centromeres, may never be fully finished.
All right. Now, the private approach was a little different. And so what they did was that rather than going through and figuring out all the genetic markers and laying out the physical map, they skipped those steps and they said, okay, we've got computers that have pretty sophisticated software. So we're going to do what's known as the whole genome shotgun approach. And so all they did was they cut the DNA into overlapping fragments that each fragment was short enough for sequencing. Okay. They would then clone the fragments and then sequence them. So they do PCR, whatever, either a plasmid or phage vectors, and they would sequence each fragment. And then once they had the sequences of all these fragments, they would stick those sequences into a computer, and the computer would order the sequences into the overall sequence, right? So they're using the software to say, okay, here's where things overlap. We can say with like 99% certainty that it's going to go right here. And the shotgun approach is much faster. It works much quicker because you don't have to do those other two steps. So you're able to go through things much quicker. And shotgun sequencing is what is done now for most genetic sequencing. Whenever it's done, it is going to be a shotgun approach because, again, it's much faster. And fidelity has increased tremendously since kind of the early days when they did this. Not saying that it wasn't an accurate method at the time, but it just is much more accurate now. So again, I got a little video so you can see what's going on with this.
Shotgun sequencing is the method that was used by the private Genome project. Shotgun sequencing requires multiple copies of the genome, which are effectively blown up into millions of small fragments. Each fragment is then sequenced. The small fragments are assembled using an immense amount of computer power to match overlapping sections. The drawback of this method comes when dealing with repeat sequences. Often there is no way of knowing how long the repeat sequence is, or in which of the many different possible positions the fragments overlap. Even the incredibly powerful software used to shotgun sequence the human genome couldn't cope with this. So Celera, the private company which relied on this approach, had to use the public data to fill in the gaps left by the repeats.
Okay, so you can see that when we have those kind of many repeating segments, actually just in this picture right here, you can see that the computers kind of have a hard time figuring out exactly where things go, and a large portion of our DNA is actually made up of those things. But so anyways, another goal of the Human Genome Project was the reduction of the cost of sequencing. The Human Genome Project cost over $3 billion, and at the end of that time, it would be a cost of about $100 million to have one human genome, so one person have their entire genome sequenced. At this time, it is down to about $7,000, and it is continuing to decrease, which is pretty phenomenal. Considering just how expensive it was even 20 years ago. All right, bioinformatics. So all of this data that we collected through the Human Genome Project and all the sequencing that has been done subsequently has been put onto publicly available websites. And these websites you and I can access. We're actually going to do a lab where we are going to take a look at these websites, I believe next week. And so the main website is the NCBI, which is the National Center for Biotechnology Information. There are websites in other languages for for those who speak other languages as well. It was created by the NIH, which is the National Institutes of Health. And some of the just kind of quick hits on some of the main databases that are available. There's GenBake, which is a database full of genes, and other sequences. And you can look up specific genes, and you can get the exact sequence of those genes. You can see those genes in different types of people. It's really rather interesting. We've got BLAST, which is the basic local alignment search tool, which is you can give it a sequence of DNA, and it will search all of the sequences of DNA that it has in its entire database, and it will compare them and see which ones match the most, which is a really cool tool, pretty phenomenal. And then we've got the Protein Data Bank, which is a database of 3D protein structures. So you can get a protein structure with amino acids and see, okay, how is this going to fold? What does it do? What are the different parts of it? How does it look? And that's going to, help determine kind of what its function is. So some really cool stuff out there. Just kind of an overview of genomes after some of the research that was done. We kind of have a general idea of some of the things between the different types of organisms and what their genome is generally going to look like. You can see that bacteria and archaea, their genomes are much smaller than eukarya. The number of genes is much smaller. Gene density is much higher in eukaryotes, whereas in prokaryotes have a high gene density. Eukaryotes have a low gene density. And the larger the genomes get, generally the lower the density. Bacteria don't have introns. Archaea do have some introns, but not nearly as many as eukaryotes. And then other non-coding DNA. Bacteria and archaea have very little non-coding DNA. Eukarya have lots and lots of non-coding DNA. We're going to take a look at that in just a minute. Okay, now, because of all this information that we've kind of been able to pull together, it's much easier to look at the totality of how all of the proteins and or genes are going to interact within an organism. So we can say, okay, these are all the proteins that interact within this particular cell. Let's compare them all. Let's take a look at all the genes and see how they're kind of interacting with one another. How is the expression being affected? And we can do that again because we've got these computers and systems that can help us take a look at this work, whereas for maybe one person, that would be much more difficult. All right, here's an application of that systems biology. It's the Cancer Genome Atlas, and this is a collection of different information about different types of cancer. So what they'll do is that they'll have clinical information from a participant in a program, and then from that person, they will collect information about the weights of a sample portion. So like of a tumor, they'll have a tumor. They'll have some histopathology slides. So what that means is that they will have, images of the actual cross sections of the tumor, so you can see the cells and what they look like. They'll have molecular information, so they will grind up the tumor and they'll look at what mRNAs are in there, what miRNAs are in there, what proteins they find. All kinds of different things. It's really a wide variety of information. And then what they also do is that they'll take normal tissue samples for comparison. So they'll say, okay, here's a cancer sample from the individual, but here's also a normal tissue sample, so we can compare them and say, okay, is this a germline mutation, or is this a mutation that is just occurring in the cancer cells but is not present in the rest of their body? So again, just a wide variety of information where, we can take a look and see, okay, what is the total expression that's going on in these cells, in this person as a whole, and be able to learn a lot of information. Those of you who are going to become either doctors or researchers, you all will most likely use this tool if you're going to be studying cancer. So really cool, interesting stuff. Okay, so we did the Human Genome Project. We found out some things about the human genome. We're going to talk about some of those here. So this is what the human genome looks like when we actually break it down. Only 1.5% of the entire human genome actually codes for proteins or gives rise to RNA or tRNA. That's a very low percentage. Really kind of interesting. And that makes us wonder, okay, what's going on with the rest of the genome? And what we actually find is that there are places in the genome that are not in the exons that are highly conserved across species, which means that they must be important. Otherwise, why would they be highly conserved? Why would animals be having them between mice and humans, right? It doesn't code for a protein, so it must be important somehow. About 24% are introns and regulatory sequences, which we've kind of been taking a look at. And then we've got 44% that are repetitive DNA that can include transposable elements. We'll talk about that in just a little bit. We've got 50% that is repetitive DNA unrelated to transposable elements. And then we've got unique non-coding DNA. When this information originally came out, people didn't understand that all of that extra 1.5%, the rest of the 98.5% was actually useful, so they called it junk DNA, and you still might hear that term some. But again, that junk DNA actually really has a lot of stuff going on. So transposable elements, these are pieces of DNA that will actually move around your genome, and these were first proposed in the 1950s. And again, they're DNA that is going to move from one location to another. We can just have a normal transposon. The transposon is going to be copied, and then that mobile DNA is going to be inserted into the DNA later on. The reason why I've got a picture of corn here is that you can kind of see that we've got different colors going on in the corn, in the corn, and that is due to transposable elements, again, that were originally proposed back in the 1950s. Now, people didn't believe McClintock, Barbara McClintock is the lady who did this. People didn't believe her when it first came out, but she did eventually get a Nobel Prize for her work. So we have transposons. We also have retrotransposons, and these code into RNA, and that RNA then codes for reverse transcriptase, which turns the RNA into DNA, and then that DNA is reinserted back into a new location. Now, this is kind of interesting because this is very similar to what retroviruses do, and so retrotransposons are thought to possibly be the ancestors of retroviruses. Examples of retroviruses being HIV, right? All right, so some other elements. So we've got ALU elements. These are retrotransposons that are found in all primates. So these are unique to just primates. We've got simple sequence DNA, which can be single tandem repeats, which are usually 2 to 5 base pairs long, and the whole sequence can be 500 base pairs. So you've got repeats of a 2 to 5 nucleotide sequence that can be up to 500 total long. We have large segment duplications. These can be thousands of base pairs long, and then large segments, so you can have hundreds of nucleotides in a row, and those are just repeated. And then we can have our unique non-coding DNA, which are gene fragments and pseudogenes. Pseudogenes are genes that have had so many mutations that by this point they no longer actually produce usable proteins, but they still code for genes, right? They still go through the transcription translation process, but the proteins don't do anything. Just a brief aside, the simple sequence DNA and the large segment duplications, these are a lot of things that are considered the RFLPs, VNTRs. When we talked about those when we do genetic testing, these are those sequences of DNA that are used for genetic testing. All right, so genome evolution. So we can compare and analyze genomes to look for evolutionary patterns. We can take a look at gene families and chromosomal alterations. So gene families are genes that have a similar structure or sequence. They can either have an identical sequence or non-identical sequence. The ones that have an identical sequence, these are going to be found in the same place of DNA. So it's going to be the same DNA nucleotide sequence that is found repeating several times in a row. For example, rRNA is a identical sequence family, and you find many copies of the rRNA in a row on the chromosomes. And you can kind of see this is the RNA transcripts that are being built off of the DNA, and you can see that you have a lot of those ribosomes, which makes sense, right? You want a lot of ribosomes being produced at a time, okay? We also have the non-identical sequence families. These are where we have a alpha globin and beta globin, right? They are very similar to one another in terms of what their construction and all those types of things, and they have evolved from a single place. We'll take a look at that evolution in just a minute. We can also have chromosomal rearrangements, so chromosomes can fuse together. So the human chromosome 16 has blocks of DNA sequences that are extremely similar to 4 different mouse chromosomes. So what we believe is that the mouse chromosomes actually fuse together into a single chromosome in humans, or by the time we get to humans, which is the human chromosome number 16. We can also have movements of the chromosomes during crossing over. I don't remember if you remember, we talked about translocations. So where a piece of a chromosome moves from one to another, well, if it gets rearranged, it's can be non-lethal, right? And what we see is that maybe that individual passes that on, and that it begins to spread throughout a population. Eventually, all of that population has that translocation. You can kind of see how evolution has occurred over time. Maybe you compare 2 different populations. In one population, there is no translocation. In another population, there is a translocation. You can see maybe where they split. So this is kind of how this works. And when we get into evolution, I think it'll kind of help you be able to see this a little bit more. But just based off of what you kind of already know about evolution, this should make sense, right? So we have an ancestral globin gene. That global gene gets duplicated. You then have mutations that occur in both copies of the gene, right? But then there's a transition to different chromosomes. And now that those genes are on different chromosomes, which is true, the beta alpha globin genes are on different chromosomes, they now are going to be kind of, having either mutations, duplications, whatever it is, that is going to lead them to have a different structure. They're similar, right? They have came from the original gene, but they've evolved separately. And so that's a really good way to kind of take a look and see, okay, where has this evolution been occurring? Which organisms have these different elements? Which is, I think it's really cool to be able to take a look at the DNA and actually be able to see genes evolving over time. Now, the other thing we can do is that, because we have all this information, we can compare genomes. What we can do is we can look at DNA and proteins to see the differences and similarities, and this is the most important piece of establishing evolutionary relationships that we use today. We take a look at DNA and protein sequences, and we see, okay, what amino acids are there, which ones are more similar to each other, which ones are more different, and we therefore establish evolutionary trees. And so what we find is that we have bacteria, which are the most close to the ancestral bacteria. They were the things that kind of split off first, and then we have archaea and eukarya, which split off from each other, and then within eukarya we've got mouse, which is less evolutionary related than humans and chimpanzees. And again, we knew these things beforehand for most of these types of organisms, but the DNA and protein evidence kind of confirms these things, and there has been some new light that has been shed in certain places, and we'll talk about that when we get into our evolution unit, which is coming up next. So thanks for watching. I hope you have a good day, and I'll see you next time.